How are T-cell lymphoma cells identified? Have you ever wondered how doctors identify T-cell lymphoma cells? This process is quite fascinating and involves several advanced techniques. T-cell lymphoma cells are identified through a mix of molecular, immunophenotypic, and genetic methods. These techniques help detect the unique characteristics of these cells and their clonal nature. One of the primary methods used is flow cytometry. This technique analyzes specific surface markers on T-cell lymphoma cells that differ from normal T cells. By using antibodies that target proteins like CD3, CD4, CD8, and CD26, doctors can determine the presence of malignant cells. For instance, in certain types of cutaneous T-cell lymphomas, malignant cells may show a loss of CD26 or unusual expressions of T-cell receptor V-beta chains. Flow cytometry helps isolate these malignant cells based on their unique surface protein profiles. Another important method is T-cell receptor gene rearrangement analysis. T-cell lymphomas arise from the clonal expansion of a single T-cell, which means their T-cell receptor genes show specific rearrangements. Polymerase chain reaction can detect these rearrangements, but next-generation sequencing offers even greater sensitivity and specificity. This advanced technique can identify malignant T-cell clones, even in early stages of the disease. Next-generation sequencing is also used to detect somatic mutations in genes commonly altered in mature T-cell lymphomas. By targeting genes like TP53 and JAK1, this method can provide crucial information for diagnosis and treatment decisions. It is particularly valuable when T-cell receptor clonality is unclear, as the presence of specific genetic mutations can confirm malignancy. In addition to these techniques, single-cell RNA sequencing is gaining traction. This method provides detailed insights into the gene expression patterns of T-cell lymphoma cells. By analyzing individual cells, researchers can distinguish malignant T cells from benign inflammatory cells, helping to understand the tumor microenvironment better. Finally, immunohistochemistry plays a vital role in identifying T-cell lymphoma cells. Tissue biopsies are examined using immunohistochemical staining for T-cell markers and lymphoma-associated antigens. This helps localize and confirm the presence of malignant T-cell populations in lymph nodes, skin, or other affected tissues. In clinical practice, these methods are often combined to accurately identify T-cell lymphoma cells. This comprehensive approach helps confirm diagnoses, assess the extent of the disease, and guide treatment decisions. Flow cytometry and T-cell receptor gene analysis are foundational techniques, while next-generation sequencing and single-cell methods provide deeper molecular insights, especially for complex cases. Always consult healthcare professionals for personalized medical advice regarding T-cell lymphoma and its identification.